Hi friends, Namaskar. Today we are going to discuss about the cleaning validation. What is the role of cleaning validation in the pharmaceutical industry, how it is useful in the pharmaceutical industry, and how it can be done. There are certain parameters, there are certain limits to carry out the validations. Validations means to verify the established standard and to meet the established set parameters. Means the result should meets the criteria with your established specification. That is validation. So in cleaning validation, what is the role of cleaning validation in pharmaceutical industry? First of all, we will understand what is mean by cleaning. Cleaning means when if we manufacture one product on any machine, that machine is fully contaminated or fully occupied with the dust and particles or the powder with the existing product. So if we want to manufacture another product on the same machine, we have to clean the machine to avoid the contamination. Because suppose if we manufacture first time metformin and second we are going to manufacture paracetamol, both the products have different therapeutic category and different therapeutic use. So the intention behind that, the product should not contaminate, and that traces or the particles of previous product should not go to the next product, and it should be safe for the human consumption. Because in pharmaceutical we are dealing with the human life, and the drugs is utilized for the human consumption. So that is our role, to make the medicine safe, effective, and quality parameters. To maintain these all the things, we are going to validate the machines for the cleaning procedure. So cleaning procedure means there may be a cleaning procedure, established procedure, standard operating procedure, but we have to verify whether that procedure is established and meeting our predefined standard or not. So for that purpose, what we are doing, we are cleaning the machine with certain detergents, waters, okay. And once it is machine cleaned, that machine should be verified with the visual inspection. There is a criteria, visual checks. Means if you found out that machine is cleaned thoroughly by visually, it means machine is cleaned. But that is the visual acceptance. What about the technical acceptance or the chemical or instrumental acceptance? So there are two methods, rinse method and swab method. We are doing the cleaning validation with these two methods, rinse method and swab methods. Many times the equipment parts is very difficult to clean and difficult to touch. So in that scenario, we are doing indirect method, that is the rinse method. We are adding some solutions, water, and taking the rinse and sending into the quality control department to verify the acceptance criteria or the presence of molecule in that particle or in that water, clean water, or establish that solution. Rinse. Later on, second method is swab method. For swab method, we are using cotton wool. That should be, we are taking the swabs from the internal parts or the contact parts of the products of the machine. Wherever the products contacted with the machine, that parts is considered for the swab method. Okay. And so in this way, there are two methods, swab method and rinse method. We are performing the cleaning validation with this mechanism. Third part is visual check. Visually also check whether machine is cleaned or not, but that is the physical acceptance. When we take the sample with rinse method or swab method, it is sent to the quality control department. They checks on the HPLC whether any peak is identified there the previous product. If in that rinse or the swab sample previous product traces is observed, it means the cleaning is failed. But there is a certain criteria, certain limits which can be accepted, like 10 ppm criteria, means parts per million, 10 parts per million. So that may be one of the criteria, we can accept it. Also there are certain calculations like ADE, PDE, and MACO. ADE means acceptable daily exposure, PDE means permitted daily exposure, and MACO is the maximum carryover allowable. So, in this mechanism, with the help of these calculations, we are performing the cleaning validation. How it is calculated, that we can discuss in another video with the help of calculations or PDF. I will provide you. If anybody needs, can comment it. I will provide that PDF as well. But here we will understand what is the process and how it is to be done. So, with the help of ADE, PDE value and MACO, we are calculating the cleaning validation calculations, and the we fix the acceptance criteria. Sometimes worst case is considered. Worst case means depend on the potency of the product, difficult to clean, and it is more contributing to the hazardous or toxicity. So that is called the worst case product. Many times handling of worst case is difficult and it is properly not cleaned. That time we have to consider our cleaning method, that is the robust method, cleaned properly and verify all the results on the HPLC. So quality control area plays a major role in this cleaning validation. They are taking the samples, they are taking the rinse swab methods, they are doing all the analytical testing. So we have seen the physical verification, analytical verification. There is third one is the microbial. What about? Because if any microbial growth in your area, it means it will contaminate the product. So there are certain criteria, there are certain limits to establish the microbial specification. However, there are two methods to handle the equipment, like uncleaned equipment and cleaned equipment. For uncleaned equipment and clean equipment, we have to establish the hold time. Hold time means after manufacturing the product, for how many days you can keep the equipment in unclean condition. So mostly people are doing 5 days, 7 days study. After that study or during the study, they are taking samples first day, third day, 5 day, fifth day, seventh day, and sometimes 10th day also for chemical testing as well as the microbial testing. If your solution or if your equipment is passed in 10th day study, you can establish a stringent criteria, that is the hold time for 7th day. So means after every 7th day you have to clean the equipment. Later on there is a clean equipment hold time study. Clean equipment means suppose we have cleaned the machine and it is kept as it is, but it is not used for the 1 week, 2 week. So in this scenario, what we have to do? We have to establish the whole time of cleaned equipment as well. For that cleaned equipment whole time study, what people are doing, same process, verification of the cleaned equipment will be hold as it is. Verification will be done through the protocol. There is a protocol for cleaning validation, whole time study of the equipment, cleaned equipment as well as the dirty equipment. In this scenario, they are doing first day, third day, fifth, seventh, 10th day, like a same. If suppose 10th day results are okay with respect to chemical and micro, they can establish a stringent criteria for 7th day. If suppose 10th day fail, they can consider 7th day, but based on they have to establish 5th day or 7th day both. But it is depend on the decision of the manufacturing site and quality peoples. But we have to consider the stringent criteria. So in this scenario, we have to face the cleaning validation. We have to perform the cleaning validation. Cleaning validation performs the major role because the drug manufacturing, it should be safe for the human consumption. With respect to safety, efficacy and quality parameters, we have to maximum control and monitor the products. However, this cleaning validation also established for the suppose same product and same equipment is there. So if product is same and equipment is same, there we can do the bracketing standard as well. Suppose same equipment having 200 to 500 kg batch size, But if you have established 200 to 500 kg batches, means if 300 kg batches is there, you can use the same equipment. There is no need to perform another cleaning validation. So that is, we can say that bracketing. In this way, if suppose product lower strength, higher strength we have done, and on the basis of this we can also do the bracketing. So in this cleaning validation parameters, we have seen how to clean, which mechanism, which methods we have to use, like rinse, swab, and visual checks also. What is the whole time of dirty equipment? What is the whole time of cleaned equipment? However, we have to focus on the training of the people. Whenever the people joins, we have to give the training on the base of standard operating procedure. Validation master protocol is there, and basis on that we have to give them training. Also, on-job training to be given to the people as and when required. If any change in SOP, if any change in cleaning mechanism, if new product comes, if the equipment change, all this scenario we have to train the people. Before starting any activity, we have to establish the people are trained on that particular activity. That training records to be recorded, maintained for a whole lifelong or as per the documentation practices. However, all these activity cleaning validation to be performed with respect to the analytical or cleaning validation protocol. In cleaning validation protocol, your scope should be clear, your objective should be clear, what equipment you are going to validate, what is the cleaning agent, what is the cleaning mechanism, what is the concentration of cleaning agent, how it cleaned with cleaning agent, detergent, or potable water or purified water. All these things should be clear cut mentioned. Basis on that, equipment whole time study should be mentioned properly. Equipment size, critical parts, difficult to wash, means uh, suppose unable to reach, difficult to reach, okay. Equipment contact part with the product, all products, all equipments photograph should be taken and mentioned into the protocol. During audit, when we discuss with the auditor, that time equipment is not in front of our eyes. Rather than we know, but auditor doesn't know what type of our equipment is there. So we have to take the photograph of equipment, we have to show the contact parts with proper labeling, with proper arrow marks. All these photographs should be in protocol. Protocol should be prepared by user department, should be reviewed by quality assurance, should be approved by quality assurance. In some of the cases, quality assurance prepare the protocol, prepared by, reviewed by, approved by, and CFT, cross-functional team, also should sign, like production head, production manager, quality control manager, microbiology manager, if required, engineering head. This is the CFT, means cross-functional team. They have to participate in this activity and establish the cleaning validation method for the sites. Many times we are going through the mechanism, what is the cleaning, and most of the time auditor most focuses on the cleaning activity, maybe area cleaning, equipment cleaning, or new equipment came, establishment of procedure, how you test the rinse sample, all these. So there are different criteria we have to follow. We have to establish our cleaning validation procedure in the pharmaceutical, because contamination and cross contamination is never accepted and permitted in the pharmaceutical industry, because it will impact on the patient's safety. So we have to be very careful about the patient safety. Patient should be safe. Previous product traces should not carry over to the next product. How it can be controlled? With the help of cleaning process only, cleaning validation process only. Once your process is validated, established, you can use that procedure lifelong. If there are certain changes, again you have to establish the procedure. So this is all about the cleaning validation, how it can be useful in the pharmaceutical. For considering the patient safety, we are doing the cleaning validation in the pharmaceutical industry. Hope you like this video. If you like, comment it, share it, subscribe it, and if you have any desire to learn more videos, more topic, you can comment it so that we can come up with the new video for you. Thank you.